Good evening, everyone. I hope you can hear me here and on Zoom. My name is Brett Stacy. I'm the Division Chief of Pain Medicine here at the Department of Anesthesiology and Pain Medicine at the University of Washington in lovely Seattle, Washington. It is my pleasure to welcome Dr. George Mashour to give the 38th Annual Gunn Loke Lecture tonight entitled The Psychedelic Renaissance. If you're interested in CME, which can be free and obtained, follow directions on the very next slide. Boom, right there. So I'll give you a minute to take that in. As I say, a couple other things. First is, we have a Gunn Loke lecture because of the generosity of Chan Gunn and his wife, Peggy. Over the last several decades, they have funded this lecture series, which has allowed us to bring renowned leaders from around the world to the UW campus to meet with faculty, staff, colleagues, students, trainees at all levels, and interact. It is such a valuable experience. We really appreciate that. Next thing to keep in mind is we are in the William Foege Auditorium. William Foege was a UW medicine graduate who went on to head the CDC and was one of the people that pushed for and made it happen, the eradication of smallpox eradication. He died last week, so I think we just should take a moment to remember him. Okay, so now on to our guest, Dr. Mashour, who, when we invited him, said, you know, I'm not a pain guy. But the truth is, we know that neuroscience is pain, and pain is neuroscience. And you can't understand pain without understanding neuroscience. He is a neuroscientist, it turns out, and a neuroanesthesiologist, and he knows what he's talking about. So has a pretty illustrious career. Currently, he is a Senior Associate Dean for Faculty and Faculty Development, Founding Director of the Michigan Psychedelic Center, Professor of Anesthesiology and Pharmacology at the University of Michigan, my alma mater. And wait, Dr. Sullivan, where are you? Oh, wait, any other Michigan Wolverines in the crowd? Yes, okay, there's 3 of us here at least. Okay, no, don't say that. Okay, but Dr. Mashour got his BA at St. John's, got his MD and PhD at Georgetown, had a Fulbright, was a Fulbright scholar, did his anesthesia training at MGH, and then moved to Ann Arbor. And in Ann Arbor, he quickly progressed to multiple levels of administrative success and research success with nearly 20 years of continued funding, several hundred publications, including more than 230 peer-reviewed papers, multiple active grants at the present time. And he does interesting things. Consciousness, psychedelics, these are really cool things. And these are things that we know are going to be in the future of medicine. So I'm very excited for the talk tonight and hope you all are as well. And afterwards, please feel free to linger and ask some questions. And welcome, Dr. Mashour.
Thank you very much, Brett, for that warm and generous introduction. Thank you for the invitation to speak. Many thanks to the Gunn family. Thank you all for attending here and virtually. It's really a great pleasure to be here. This is my 3rd time lecturing at the University of Washington since I've been a faculty member, and it's really been a delight to be back. I feel very honored. I also feel very unworthy to give this lecture because I did push back, said, I'm not a pain physician. I'm not a pain scientist. I have not made contributions to the field of pain. And I've seen the list of past lecturers, Melzak, Wall, Fields, Bushnell, Basma, and the list goes on. I don't deserve to be on it, but I'm very thankful to be a part of this. And as was noted, although I don't study pain as a primary focus, I am deeply interested in consciousness. And I'm interested in pain as a state of consciousness, pain potentially as an evolutionary driver of conscious experience, pain and nociplastic pain in particular as a potential disorder of consciousness. And I think that the fields of consciousness science and pain neurobiology and clinical pain can have a very rich dialogue. So why study psychedelics, aside from the fact that they're cool, which I think it is a cool field. And I have put up a quotation from Dr. Stanislav Grof, who's a psychiatrist, really a pioneer in psychedelic psychotherapy. And he wrote, psychedelics used responsibly and with proper caution would be for psychiatry, I'd broaden that and say for the study of the mind and the brain, what the microscope is for biology and medicine, or the telescope is for astronomy. So psychedelics are a very powerful lens through which we can see key aspects of brain function and cognitive function. Where they differ from these other lenses is that psychedelics not only reveal, but potentially can also heal. So that's why I'm interested in psychedelics. I spent the first part of my career focused on studying anesthetics as tools to modulate and understand various levels of consciousness, global states of arousal. Psychedelics help us understand the content of consciousness. I also wanted to share why we use the word psychedelic. Traditionally, in the 1950s, these drugs were referred to as psychotomimetics. And actually, it was Ralph Gerard, who was at the University of Michigan, founded the Mental Health Research Institute, who coined that word. So these drugs were thought to mimic psychosis. But as people came to understand their properties, they felt that that was not quite appropriate. And there was a dialogue between Aldous Huxley, the writer, and also Humphrey Osmond, a British psychiatrist. So Huxley said to present an alternative to psychotomimetic, to make this trivial world sublime, take 0.5 gram of phenerethyme. And that's what he sent to Osmond. To which Osmond replied, to fathom hell or go angelic, just take a pinch of psychedelic. And even though Huxley was the famous writer, Osmond's term psychedelic ultimately won out. And this derives from the Greek, and it combines psyche or psyche, soul, mind, and delos, to be revealed. So these are drugs that are revealed in the mind, our mind manifesting. Now, psychedelics, of course, have been used for millennia by indigenous peoples in religious and spiritual contexts and ceremonies. But when we think of them in the Western world, in the United States, we often think of scenes like this from the late 1960s, where psychedelics had a major influence on music, on art, on architecture, on political sensibility. And you can see some of these images. But in the early 1970s, much of this died off, and a lot of research in psychedelics died off after they were made Schedule I drugs. Also keep in mind, in the early 1960s, LSD was legal. It wasn't until the mid-1960s that it was illegal. So in the 70s, things went underground. There were still a few laboratories working on psychedelics, but kind of the dark ages in recent history for psychedelic research and therapy. And then things started to change about 15 years ago. And really my main goal today is to try to help set the stage and frame the moment in which we're functioning as physicians and scientists and academics. And also to let you know the state of play in pain research, because while psychedelics are enjoying a rebirth, I think the interface of psychedelics and pain are still in the birthing process. And I'm going to bring up data, some of which has come out within the past week. Like I haven't even had time to read the articles. It is dynamic and also unclear. So what are the forces that have coalesced to create this psychedelic renaissance? It's started to appear in the popular literature. So I'm sure many of you are familiar with this book by Michael Pollan. It was subsequently a series. There were centers at major academic institutions that were founded, focused on psychedelics. Now, I founded a Center for Consciousness Science in 2014, and we would host various symposia, including on altered states. And in September of 2019, we were hosting a symposium on psychedelic neuroscience and therapy. And when this news hit that there was a $17 million donation to found a new psychedelic center at Johns Hopkins, no less, it was a big event. And that has proliferated, as has the research. Now, this is an article from 2020, and you can see what happened between 2010 and 2020. This is what's happened since then. You can see the past 5 years, this spike in publications. And it's not just high quantity, there's also high quality. Major randomized trials that are being published in the New England Journal and Lancet, major scientific articles that are being published in Nature, including this one. And I highlighted this because it focuses on a very interesting and emerging topic. Here it's referred to as the non-hallucinogenic psychedelic analog, also sometimes referred to as psychoplastogen. So these are drugs that are thought, based on preclinical data, to have the therapeutic efficacy, and they're analogs of psychedelics, without the trip. And there's an emerging class of drugs and drug discovery efforts in this area. And I think it's really important to highlight because, and I'll describe one clinical trial on psychedelics. It is timely, it's costly, it's labor intensive. And the notion of having a therapy that somebody could take, go to work, have access to it on a regular basis could be a game changer. The other reason why I want to highlight it is in situations where efficacy is promising, such as depression, for example, it's not yet known how psychedelics are working. Is it just having the drug in the brain that's inducing neuroplasticity, which both classical and non-classical psychedelics do? They induce dendritic sprouting and neuroplasticity. Or is it the experience that the drug is evoking? There's something about the phenomenology and the first-person experience that's having a therapeutic effect. Or is it the therapy that goes along with the drug? The drug perhaps is creating certain conditions, but the therapy is what's really efficacious. Or is it expectancy and placebo effect? Or is it some combination thereof? And that's unclear right now. And of course, it's very difficult to blind people when they're getting a psychedelic. And I'll talk a little bit about that later. The other thing that's changing is policy and law with respect to psychedelics. And you can see the map here, and you can see your state and mine, where there have been local reforms. So there are certain states where it is legal at the state level, not at the federal level. In Seattle and in Ann Arbor, where I'm from, entheogenic substances, psychedelic mushrooms, for example, are decriminalized. It doesn't mean they're legal. It means that they're deprioritized and they're not prosecuted. And this is important. It's been important and likely has followed the course of cannabis. And I know when I was growing up in the 70s, they talked about pot as the gateway drug. And in this case, it's kind of true. You know, a lot of the playbook for cannabis has been employed with psychedelics. So that's the context of the psychedelic renaissance, and that was the context within which I had founded the Michigan Psychedelic Center in 2022. Originally, it started off as the Michigan Psychedelic Collaborative. I didn't want to do anything official, see if there was an organic community that would emerge, and it did across campus. A lot of interest across the medical school, but also various centers, and you can see them up here. Nursing, public health, social work, where there's a student organization that we sponsor. The School of Music, Theater, and Dance. The chair of the dance department has published books on psychedelic rock. So I've enjoyed communicating with them related to psychedelics. So the interest has grown. It's still a small group, but it's mighty, and lots of disciplinary diversity. And you can see the tagline, revealing the mind. Remember, that's the Greek origin of psychedelic, with rigor and responsibility. And this is something that I emphasize over and over with our team members, with the media when I'm interacting, is that we need to be critical and self-critical. We need to learn the lesson of the 60s. And so my motto is, we should not stigmatize psychedelic research or psychedelic drugs. Neither should we romanticize it. It's not a panacea. We need to be critical, just as we would with any other kind of drug research, because I think it's very important, if we ever want this to cross over into mainstream medicine, that we have a rock solid, credible, unimpeachable set of data with rigorous, critical thinkers and investigators who are pursuing this. We pursue the mission across education. I've started neuroscience graduate courses on psychedelic neuroscience. We host seminars and symposia, research, which I'll talk a bit about. We connect with physicians and with patients, and also community engagement. And our mission is to advance multidisciplinary education, research, therapy, and community engagement related to psychedelic drugs and the psychedelic experience. And we've also studied non-pharmacological pathways to altered states of consciousness. So I have a number of interests in this area, but fundamentally, as somebody interested in consciousness, I want to understand how these drugs work to alter perception in such a radical way. And we study psilocybin in rodents and in humans. I'll talk a little bit about that. Psilocybin is actually a prodrug for psilocin, which is the active ingredient, if you will, dimethyltryptamine. So these are all within the tryptamine group. We've worked with data sets from LSD. We've studied DOI, salvinorin, and ketamine and nitrous oxide. There have been a number of psychedelic centers that have been founded, but I believe that we're the first to be founded within a department of anesthesiology. And so we leverage that and we lean into that. And part of that has been studying drugs like ketamine and nitrous oxide, which at subanesthetic doses are psychedelic or have psychedelic properties. So the first part of this is going to be me talking about psychedelic drug action in the human brain and some of the work that we've done. And I've approached this question, as I did earlier in my career, thinking about how anesthetics work. What makes an anesthetic an anesthetic? How can we use all these structurally and chemically and pharmacologically and neurobiologically diverse agents to achieve this common functional outcome in the operating room? Similarly, we have a group of diverse drugs that seem to elicit not the same experience precisely, but sharing a lot of canonical features of an altered state or mystical state of consciousness. And we can think about some of our, I'm going to call them classical anesthetics, and the reason for that will become clear. Drugs that, to a greater or lesser extent, work through the GABA A receptor, propofol, etomidate, and ethers. There's also a group of classical psychedelics, and they are referred to as classical psychedelics. Just as the anesthetics are grouped over here because of their action at GABA, these drugs are thought to act through the 5-HT2A, 5-hydroxytryptamine or serotonin 2A receptors. Now, given the opposing actions of anesthetics and psychedelics, You wouldn't think that there'd be any interface between these two circles, and yet there is, and that's because of ketamine and nitrous oxide. And these drugs are atypical anesthetics, and they're also atypical psychedelics. And they have these dose-dependent properties. And ketamine, for example, can evoke very similar experiences. It's got a profound dissociative effect. And ketamine, speaking of the University of Michigan connection, was first studied clinically at the University of Michigan in the 1960s. It also induces neuroplasticity. It has rapid antidepressant effects. It reduces posterior alpha oscillations, very similar to these classical anesthetics. So early on in my career, I tried to think, what's common across ketamine, propofol, and ether? Now, I've started to think about ketamine, nitrous, and these classical psychedelics. And the first study that we conducted, we did a neuroimaging study of nitrous oxide, subanesthetic nitrous, where we evaluated altered states of consciousness and imaged during administration of 35% nitrous. I wouldn't recommend it. You've got somebody in an MRI who is vomiting in a mask. It is a challenge, but we did the study, and we also had data on ketamine, and we leveraged a data set from Robin Carhart Harris, who really has been at the vanguard of the psychedelic renaissance, on LSD. And what we found across these diverse drugs, diverse in their route of administration and their molecular target, is that they all reduced within network connectivity. So a functional network, let's say the frontoparietal network or the default mode network, the correlations and functional connectivity within those networks were weakened. And that's the left side of the screen, the within network connectivity going down with nitrous, ketamine, and LSD. The between network connectivity and communication, we infer communication, is increased. And so what's happening is that you have functional brain networks that are more integrated and less differentiated. And this balance is critical. And we've studied this, and this was an article we published in 2024. Consciousness and the neural substrates that support consciousness have to have a balance of differentiation or segregation and integration. Think about your experience right now. It's multimodal, it's multidimensional, it's textured, you're feeling things, you're hearing me, you're seeing me gesticulate up here. And yet, you have one seamless experience. It's unified. In the brain, you have different networks and different regions that are processing modal and submodal information that also gets integrated. They get integrated in space, in different areas of the brain. They get integrated in time. And you can see this integration segregation balance that we've measured with fMRI. And when you are in a sedated state, sedation, sleep, general anesthesia, the brain is becoming more segregated. It's becoming more modular. There are more islands of activity. By contrast, psychedelic states are pushing brain networks toward a more integrated state. And that integrated state is thought to be associated with psychedelic phenomenology. We also looked at some common areas of connectivity because the network shifts were different across these different drugs, which I want to acknowledge. But there were common patterns of connectivity in what has sometimes been called the posterior cortical hot zone of consciousness. I didn't come up with that term, nor would I. It's a little weird. But this confluence of association and sensory cortices is thought to be really important for conscious experience itself. And so it wasn't entirely surprising, but was reassuring that these points of connectivity were shared across these psychedelic drugs. We also tested propofol, excuse me, in this study. because we wanted to make sure this wasn't just a function of a major brain state change. And in fact, there was a fourth area, the insula, that dropped out. Furthermore, the temporoparietal junction became important as we analyzed the data. The TPJ, as it's sometimes referred to, is important for multisensory integration. It seemed to be correlated with some of the psychedelic phenomenology. It's important for body ownership. People who have lesions in that area start having spontaneous out-of-body experiences. And so it was of interest, and looking at the shared connectivity between that posterior cortex and the TPJ also revealed some similarities. Now, this is an article that was published in Nature with repeated doses of psilocybin that showed functional connectivity changes acutely and, importantly, probably why it got published in Nature, persistently. And again, these psychedelics are neuroplastic. And so just to reassure you that, well, what if I gave a psychostimulant? You can see what happens with methylphenidate or Ritalin compared to the degree of functional connectivity changes with psilocybin compared to just our day-to-day functional connectivity change. That's kind of depressing, right? It's all dark, not much going on. I want to be more on that left side, but I can't. Now, in psychedelic neuroscience, there are two major areas of investigation that have been somewhat separate. One is the cellular, molecular, that fundamental biology. Another is the neuroimaging of these large-scale brain networks, as I've just shown you. And we wanted to try to bridge that gap. And so instead of focusing on the large-scale connectivity, we focused on, excuse me, this smaller cluster of voxels. And you can see kind of that representation here versus a larger scale set of analyses, excuse me. So we're trying to find a bridge between, these molecular and cellular events and the large scale events. So we wanted to focus on smaller regions. And in addition to ketamine, nitrous, and LSD, we also introduced dimethyltryptamine, DMT, and psilocybin in collaboration with Chris Timmerman, the group at WashU who published that Nature paper, and Robin Carhart Harris. And what we found, and so what you're seeing on the x-axis are various functional networks, canonical networks, somatomotor network, visual, dorsal attention, ventral attention, frontoparietal, default mode. And what you can see across these different psychedelics is that they're reducing regional homogeneity. Essentially, they're creating desynchronized states within these little clusters. And this was robust to a number of different analytic techniques. And you can see some of the overlaps of all these psychedelics with these hotspots in the dorsolateral prefrontal cortex, the medial prefrontal cortex, where there is a very high concentration of 5-HT2 receptors, and also precuneus and some other areas. Now, this was interesting, and we were looking at this in relation to large-scale functional network changes, but also trying to go in the other direction and leverage PET tracer data on receptor and channel concentrations to identify links between what we're seeing with this regional or local desynchrony and what might be going on a molecular and cellular level. And what we found, so you can see those maps above, and below is what's referred to as a dominance analysis, basically to look at an association between the receptor in this case and the local desynchrony. And what we found is that for DMT LSD, and psilocybin, there was an association with the 5-hydroxytryptamine receptor. Not always 2A, which was the case with DMT, but still in the ballpark. Ketamine and nitrous oxide, the major driver appeared to be the NMDA receptor. And neither of these findings are unexpected, but they're very reassuring. To be able to link what we know about some of the molecular pharmacology with what's going on at this mesoscale, this local desynchrony, and how that translates in a drug-specific way to changes in global functional connectivity. Now, I've mentioned, and I was talking to some people beforehand about how dynamic this space is. So this is an article that was published in Cell on January 22nd. Like, I haven't even had time to parse through it yet, but it's a study in animals looking at the effects of psilocybin and how it can induce changes in functional networks. And I just shared the graphical abstract to give you a sense of it, but there are going to be a lot of exciting data, I think, that derive from this and lines of investigation where we can better connect what's going on at the large scale network level with human neuroimaging and understanding the precise circuits and the rewiring that might happen with psychedelics. Okay. I can't give an endowed lecture in pain without talking a little bit about pain. And in fact, again, because we founded our center within a department of anesthesiology, pain was a priority. And actually, there wasn't a lot of work, even in the preclinical space. Now, there are some data and publications from the 1960s on healthy volunteers and acute pain perception, but there really wasn't much of anything that we saw in terms of a systematic assessment of psychedelics in preclinical pain models. So I'm going to describe a study which, to my knowledge and to our knowledge, really was the first to test psilocybin in a pain model. And this is a formalin model of chronic pain. And these rats were instrumented also with a central venous catheter. And I credit my colleagues in the lab for popping those lines in, and more so this high density EEG. And the reason why we do that is we want to try to create some translational bridges. And just as a quick detour, this was a study that we published last year looking at functional networks with this high density EEG montage in the rat brain, how intravenous psilocybin, again, going through the central catheter, is acutely reshaping these functional brain networks and high frequency oscillations. We just published something in the Journal of Neuroscience on intravenous dimethyltryptamine. And again, you can see those points of connectivity lighting up. So that was just a quick digression. So the animal gets an injection of formalin in one paw, and this becomes centralized, and they start having sensitivity contralateral to the injection site. So this was the work of Nick Kolbman, who got his PhD in pharmacology at the University of Michigan. He's now a postdoc on a T32 in the Department of Anesthesiology at the University of Pennsylvania. And this is the key finding. So this is the assessment for mechanical hypersensitivity in the ipsilateral paw, the paw ipsilateral to the injection. The lower the threshold, the more sensitive. The higher, the less sensitive. So you can see where they're starting out at baseline. And then you can see that hypersensitivity after the formalin injection. This is the course of sensitivity for the animal that had the saline injection. This is the course of activity for those animals that had psilocybin This was a single dose of psilocybin. And what's notable here is that you're getting a durable effect. So the half-life of IV psilocybin is a couple of hours, but you're still seeing an effect at 28 days. Also speaking to a central mechanism, the contralateral paw actually has a more robust finding. It doesn't have that inflammatory local nociception going on. And you can see, again, an effect of psilocybin that's durable. Less so with thermal hyperalgesia. So Nick is working on some of the mechanisms of this toward the latter part of his PhD. These are unpublished data. In this situation, both cohorts have had psilocybin, but one cohort in the black circles has also received pre-treatment with a microinjection of M100-907, which is a selective 5-HT2A antagonist in the insula. And we focused on the insula because it appears to be important for chronic pain. You can modulate the insula and induce pain behaviors. And what you can see here is that the blockade of the 5-HT2 receptor in the insula really seems to attenuate the therapeutic effect. So you can see the therapeutic effect in the red squares, but that's attenuated, and that's looking more like the baseline if I go back to this graph. Okay, so this was, the original finding published in 2023. And then there have been a series of papers that have come out in 2025. This article, although I say rodent model number 2, there are actually a couple models here. One is a chemotherapy-induced neuropathy. The other was an inflammatory stimulus, a complete Freund's adjuvant. And I'm just showing you the data from the neuropathy. These animals had IP psilocybin at 1 mg per kg, 0.3 or 0.1. And you can see in the blue and the red, 0.3 and 1. After injection, again, you're going higher, you're more resilient, you have a higher threshold, you're less sensitive. So you can see a durable effect out to 14 days and also with cold hypersensitivity. And this was mediated, again, by 5-HT receptors. Then there's this beautiful paper published in Nature Neuroscience by Joe Cichone at the University of Pennsylvania. Again, I refer to a single model, but there are two. A spared nerve injury model of neuropathic pain and also complete Freund's adjuvant. And what you could see down here, again, these are the same interpretation. Lower means more sensitive, higher means less sensitive. What was interesting here is that they didn't give the psilocybin, and it was injected IP, until day 27 after the pain stimulus was induced. So the pain was happening for weeks, and then you can see this dramatic reversal in both models with psilocybin. Again, 5-HT2 mediated, also showing some changes in anterior cingulate cortex. So that was in November. In December of 2025, another formalin model, and basically similar findings. This was in rats as opposed to mice, the last two. And you can see that psilocybin is attenuating the effect, but when you pre-treat with volanerancerin, again, a 5-HT2 blocker, you attenuate that effect. So we published our finding originally in 2023. Multiple studies have come out that have reproduced this, if not precisely, then with different models, different species. But in the past week, again, this is an emerging dynamic literature. Boris Heifetz from Stanford published an article in Nature Communications showing no effect of psilocybin. multiple different models. Again, same interpretation, this all withdrawal latency. And you can see all these conditions involved different doses of psilocybin, no improvement. And I don't know what to tell you about that. I mean, it's easy for me to differentiate this from our study because this was in mice, we used rats, this was intraperitoneal injection, we used intravenous injection. But this is something that's going to need to be parsed through and figured out. So again, when I said this might be the rebirth of the psychedelic movement, I think the work in pain, this is still in developmental stages, and there's still a lot to figure out. I also wanted to mention, you might look at this and say, well, maybe they injected duds or blanks, like this wasn't working. How do you know this is effective at all? We can't, you can't ask the mouse, like, what's it all mean, man? Like, are you feeling, right? You can't assess whether they're tripping. So what happens is, and I was actually just talking to Dr. Brukas about this, there is this behavioral phenotype of this high frequency head twitch that happens when you deliver a classical serotonergic psychedelic to rodents. And that was observed here. It was observed in our study and these other studies. So that's how you know it's biologically active. These non-hallucinogenic psychoplastogens don't induce that head twitch response. So more to come. Now, there's not much to turn to in the human literature. There's some case reports, there's some work with headaches, but to our knowledge, we conducted the first in-human pilot study of psilocybin and fibromyalgia patients. You can take a look at the recruitment over here, 368 expressed interest, and we got down to 5. Okay, so it's not easy. You think everybody would be lining up to get their hands on these good drugs. But you can look the paper up, you can see the reason for exclusion. 7 were enrolled, 2 withdrawn, 1 couldn't handle it, 1 was not building rapport. I guess they weren't vibing with the therapist, which is important. But these were really labor intensive. The screening process, deep phenotyping, quantitative sensory testing, fMRI, patient-reported outcomes. We had multiple therapists assigned to each person. We had an anesthesiologist present for the dosing. So there were several preparatory sessions. A first dosing session, 15 milligrams IV, between interdosing visits and evaluations. Another dosing session with a higher dose of psilocybin, post-dose visit, and then weekly follow-up sessions, and then another round of deep phenotyping. And this really followed kind of the formal protocols of psychedelic-assisted therapy. Now, I'm not going to even, the patients reported improvement. I'm not going to ascribe any significance to that. It was not powered for that. I would say, even if there was benefit, this is just not, it's just not doable, and we have to be thinking about this. It was well tolerated. There was some transient heart rate increases, blood pressure increases during the dosing, but within the expected parameters. Headache was the primary adverse event that happened the day after dosing, treated, manageable with over-the-counter remedies. So it was well tolerated. And also I'd note there's variability. So, various questionnaires, including challenging experiences, what we used to call a bad trip. Now they call it a challenging experience. And also mystical experiences. And you can see, for example, participant 2, neither dose. They didn't have, it was not pleasant. They didn't have some transcendent experience, although they did report that there was some improvement. So that was interesting. It didn't seem to correlate with the classic psychedelic phenomenology. So lots of challenges with these trials. Regulatory issues, very costly, very labor intensive for this traditional approach. And again, getting back to the problem of how do you control for the placebo effect or expectancy when you're getting a psychedelic? And anybody who talks about this, I think, is compelled to bring up this Noth cartoon, which I think really tells the tale. Okay, right? So it's like, one group's getting psilocybin, the other's getting what? You're probably going to figure it out. Okay, now I want to go back to this because we're in this place right now. The animal literature is emerging, and Dr. Heifetz, bless his heart, dropped a bomb on us in the past week with that study. So it's not clear what's going on there. There are some ongoing clinical trials, including in fibromyalgia, low back pain, and we have to await those results. But in thinking about what are we going to do and how are we going to get more information, we have to remember in Seattle and in Ann Arbor, there are people who are taking mushrooms, and they're taking them for their pain conditions, and they're taking them for depression, and they're taking them for anxiety. And so we have also tried to leverage the community to try to understand their experience using psychedelics to essentially self-medicate. And this is a study, actually, Nick Linos, who is a PhD student with us in neuroscience, also got involved in some of this community engaged work. And again, we're trying to work on the preclinical models, the clinical trials, and the community engagement. And you can see in this survey of psychedelic users, 10% of this cohort had a chronic pain condition, and they share their outcomes. Of course, there are lots of methodological limitations, but we're trying to piece the picture together. And when I think about what's going to happen, like with psychedelic therapy, because I have people reaching out to me from all over with all sorts of different disease processes who are interested in psychedelics. They might be legal at the state level, but if they're not legal at the federal level, we're not going to be able to deliver them therapeutically. I think what we can do is to think about how we can harmonize between our current medical care and the naturalistic use. How do we work with patients who have depression or anxiety or pain who are using psychedelics without endorsing them or prescribing them formally or informally? So we're trying to engage the community. We engage this community. We get out there. There's actually a festival in Ann Arbor, which is famous for its cannabis festival called Hash Bash that dates back to the early 70s. But they started one called EntheoFest. And so we get out there. So that's Kevin Banke, who's the associate director of the Michigan Psychedelic Center. He's trained as an epidemiologist. This is me, like I'm wearing a sport coat to a shroom festival. I don't know what I was thinking there. But I gave a keynote there in 2024 to try to start the dialogue with the community. We had the newly elected sheriff who made a public statement about not prosecuting people who are using entheogenic substances. And what I think was a big step for us in 2025 is that we hosted the Global Psychedelic Survey. This started out as a Canadian psychedelic survey in 2022. There was a Global Psychedelic Survey in 2023, but we hosted it and we hold the data for 2025. This was translated into 19 languages. AI made up a bunch of flags there to try to represent that. We had 80 collaborators across 20 countries, and we got more than 9,000 respondents from 105 countries to talk about their experiences with a wide variety of drugs for a wide variety of indications, and pain is among them. So we will try to leverage this broad international community perspective as we're integrating this with clinical trial data as they come in, as well as preclinical models. So to summarize some of our work in the human brain, diverse psychedelics, both the classical serotonergic and also the atypical or non-classical psychedelics, ketamine and nitrous oxide, reduce local synchrony. They reduce within-network connectivity and increase between-network connectivity, shifting brain networks to a more integrated state. And again, connecting, I think, in a positive way with what we would expect, linking those changes in the human brain to 5-HT receptors for the classical psychedelics and NMDA receptors for the non-classical psychedelics. Single-dose psilocybin has shown benefit in multiple and diverse rodent models of chronic pain, including our own, but recent work has not reproduced those findings. We have to figure it out. I mean, I don't have an answer for you. You know, Joe Cichone did elegant work published in Nature Neuroscience using mice, using similar techniques and model that the Stanford lab did. We just have to figure out why there's that disparity. In terms of our first-in-class feasibility and safety trial, psilocybin was tolerable for fibromyalgia patients. They could get through it with only mild adverse effects, but we have to wait the results of ongoing clinical trials to really understand efficacy. And then finally, community engagement with psychedelic users who have chronic pain, I think is important and can be informative as we try to harmonize our current medical systems with the naturalistic use that is increasing as these drugs become legal or decriminalized. So many people I'd like to thank on the left, Dr. Dai and Dr. Huang, my collaborators for the neuroimaging work. And I also want to acknowledge Chris Timmerman and Robin Carhart-Harris for their very helpful collaboration. That's Nick Coleman, our former PhD student, now at Penn, Dr. Giancarlo Vanini and Dr. Dinesh Pal, who are leading that laboratory work. Kevin Benke, the associate director of our center, and Jake Aday, who took the lead both with the fibromyalgia trial, but also very heavily involved in the Global Psychedelic Survey, and various graduate students, Tony Larkin, now a postdoc, with our group, Nilüfer Puyan, who's getting her PhD. They both worked on the nitrous data, Nick Linos, whom I mentioned, Emma Hules, who has also done a lot of work in this space. I want to thank my funding sources, the NIH, the James S. McDonald Foundation for my work in consciousness, certainly the University of Michigan for their support of our center, TRIP Therapeutics that funded the basic science study with psilocybin and the chronic pain model. And finally, I want to thank you again for the kind invitation. I thank you all for being here and your kind attention, and I'll be happy to address any questions.